All right, so we're going to continue on kind of this tour through the realm of systematics. Previously, we talked about species identification, what determines discrete units of species, and how we identify between individuals and determine whether individuals within a population represent different species. So now we're going to move into, or kind of segue into, a different realm of systematics regarding how we actually observe or represent the relationships between these organisms and between different species. So this area that we're moving into is phylogenetic systematics. Phylogenetics is the study of evolutionary relationships between different organisms, different species, different genes, etc. And we're able to kind of observe these relationships or represent these relationships on what are known as phylogenetic trees. Phylogenetic trees are also referred to as phylogenies or evolutionary trees. They represent or depict, again, these evolutionary relationships among different organisms. And they essentially represent this tree-like structure that's composed of branches and nodes. So each branch represents a different species. These are your branches here, the straight lines. And then the nodes, or the sites of branching or splitting from an ancestral species. Those are the nodes. Those represents these divergence events. Now, these again represent the evolutionary relationships, the patterns of the branches, or that branching pattern of the trees allows us to observe these evolutionary relationships and kind of determine how species are related to one another and see how that fits into this scheme of life. Now, it's important to remember that phylogenies are hypothetical, right? So they are hypothesis-based. The relationships that are represented in these trees are hypotheses. They're not definitive facts, and that's because there's only so much information that we have available to our use to create these hypothesized relationships. As I was saying before, we continuously find new information or identify new species that causes us to reevaluate and reconstruct these phylogenies. So they are hypothetical. They're not set in stone. In general, when we create these phylogenies, there's a variety of different factors that are used. So these factors, we use morphological data, evidence or information from the fossil records, embryology or embryological features, genetic sequences. They just represent some of the factors, again, that we used in constructing these trees to represent the relationships. Now, those factors themselves are more specifically called characters. Characters are any observable characteristic of an organism. Again, this can be morphological, anatomical, developmental or embryological features, embryological patterns, behavioral patterns, genetic sequences, etc. Those are all different characteristics that can be used to determine the relationships and relatedness between different species, as well as determining whether individuals are of separate species. Now, each character has different traits. Traits you can kind of think of as being subsets of characters. Traits are character states or different values of a character. So, for example, brown fur and white fur are two traits of the character coat color. So you have characters, characters have different traits. Now, when we actually construct phylogenies, um, again, we use a variety of factors to kind of guide these hypothesis-based patterns. Originally, we used primarily morphological and anatomical data and information to kind of infer and create phylogenies. But as we talked about before when in talking about species, that's just a tiny component of what helps to differentiate between species. But you can't solely rest on that factor. If, as we were talking with like the biological the species concept and the phonetic species concept, by kind of restricting to these morphological, behavioral, or anatomical features, you can create flawed phenotypic trees or phylogenetic trees because you can't account for the fact that some speciation events involve no changes in morphology, right? The creation of your cryptic species or cryptic speciation. Some species will have similar traits, but that doesn't arise from common ancestry, right? Those are your analogous traits. So different things cannot be accounted for by looking at just that one factor. Today, and kind of in modern times, we have moved or changed to using predominantly genetic information in constructing phylogenetic trees because that is a more sound kind of factor to utilize. It can prevent the errors that would otherwise be caused if you were using other factors for cryptic species or convergent evolution. So genetics really kind of circumvents and mitigates the issues of convergent evolution, or allows you to detect the differences in species even though they have similar traits. So we started using genetics primarily following the dawn of molecular genetics, and it's really remained since that time. But in general, multiple factors are used in constructing these trees. Usually, kind of the first step in making a tree is using genetic sequence. So I have here kind of the several steps in making a tree. So this initial phylogenetic tree has been created from the character of genetic sequence. So genetic sequence here is the character that is being used to infer the phylogenetic relationship of the species of interest. And that creates this initial phylogenetic tree. Now down here, that same phylogenetic tree that's been created has been taken and it's now having secondary sets of characters added to it. So additional attributes are being added to the tree to further guide our understanding of the evolutionary events and also reconstruct the tree as needed. So take our initial tree that was generated by the genetic sequence. here we're overlaying the characters of flower morphology, looking at the shape and color of these flowers, and overlaying those factors or those characters onto the tree allows us to make further inferences about these evolutionary events and relationships. So, for example, we can now say, okay, this here, from this ancestor, you had the origin of orange flowers, and you had that divergence there. So, orange flowers originated with this ancestor, and we can say that so these two white flowers here come from the same ancestor, but this species is different from this species because it has a novel shape, right? So because you have the arisal of a novel shape here that is unique to this species, and that's what differentiates them. So again, you can add components and features that help you better understand these evolutionary relationships and understand kind of the origin and changes of traits that have occurred. Now let's actually pick apart the internal anatomy of these trees. So as I said, your phylogenetic trees are like trees. They have a branching structure that's composed of branches and nodes. Now the branches are these major horizontal lines. Each horizontal line represents a different species. So here, Figaro, here our species of interest are A, B, C, D, and E. Each one has its own branch, and the species that kind of correspond to each branch are usually situated right at the tip of each branch. Stop it. Now, in general, you can determine relatedness based on kind of the internal structure of the branching pattern. So you have these branches. The point that they stem from is known as the branch point. So the branch point you can think of as the fork in the road. This is the site of a divergence event that allowed you to have the formation of these two new species. Now, every branch, every branch point represents that divergence event, also represents a common ancestor. In general, species will be more closely related if they have a more recent common ancestor. Species will be less closely related if they have a less recent or more distant common ancestor. So species A is more closely related to species B because they have a recent common ancestor than A is related to C. Right? That's a more distant common ancestor. A lot of trees are rooted, meaning the last common ancestor for all of the species of interest is given its own branch. And that kind of helps set kind of the scheme or the scope of the relationships between these species and figuring out where they originally stemmed from. What was that original divergence event? Now, most trees, all the branches are kind of aligned along this vertical axis. Other trees, you don't see that. They have different branch lengths. In these kind of trees, they're known as phylograms. The branch length actually represents or is used as a measure of evolutionary time, where the longer the length, the longer the evolutionary history of that species. It also represents the amount of genetic change that has occurred. So the longer the branch, the more genetic change that's occurred for that species over the history of evolution. Now, these lengths can be typically generated by measuring the amount of differences in genetic sequence information. So based on the number of differences and changes that have occurred, you can generate kind of the length or evolutionary duration of that species. Most trees will move left to right, where lefternmost is more ancestral. As you move towards the right, you get closer and closer to present day species. Some are flipped, but in general, you have this trend where you go from less branching to more fine-tuned, more branching. As things become more and more branched, you move towards present day. So usually less branched is more on the ancestral end, highly branched is moving towards present day species. As I said before, all these trees have branch points. And those branch points, again, they're like the little fork in a road where you have the divergence of different species. And that is also known as a node. So these represent divergence events. They also represent the common ancestor. So we can say that, so here's your individual species. They can also be called taxon. So species are taxon A, B, and C. They all arose from this common ancestor at branch point 3. So this is the common ancestor for those 3 taxon, and that was the site of the divergence event. If you have 2 species, so we can call all the species that arise from these divergence events, we can call them as descendants. So species B and C are a good example of this. When you have 2 descendants that split from the same node, right? So they both split from 4. They're referred to as sister taxa. They are each other's kind of closest relatives, and they share an immediate common ancestor. And some trees, you'll see kind of a weird structure where you might have a kind of a pitchfork-looking thing, or a branch point serves as a point for the divergence of more than 2 species. When you see this kind of structure, it's called polytomy. These are essentially realms of question that we have in the scope of phylogenetics. We're not sure. We know that they all evolved from this ancestor and from this divergence event, but we don't know exactly where they go from that. We don't know if taxon E diverged from D. We don't know if D diverged from F, if they all evolved or diverged immediately from this event. It's kind of this site of unresolved pattern of divergence. So usually we'll just place them all on a single plane until we can better understand the evolutionary relationship there. You see this with monotremes because we're not sure where exactly to place them. We know that they are in Mammalia, but we don't know if they're more closely related to placental mammals or if they're more related to marsupials, but we can tell what they're kind of that they share a common ancestor with your mammals, and further an even more distant common ancestor with reptiles. So again, they're just kind of these zones of uncertainty. Sometimes you'll have what's known as a basal taxon. So in this example, taxon G is a basal taxon. Basal taxon are referred to as outgroups. So an outgroup is just a taxon that's outside of the group of interest. So all of the species that you are interested in are more closely related to one another than they are to the outgroup. You can kind of think of this as like an outlier or an outcast. But it's used often to help broaden the scope of where this evolutionary relationship fits in, or how it fits into the larger scope of life. It also can sometimes reveal interesting relationships or information or relatedness that you didn't know before. You can find that an outgroup is actually more closely related to a feature of the in-group than you had previously thought. So those are some of the main internal features. Again, we're slowly going through the anatomy. We're going to go into kind of the different types or different patterns that you can see now, and then we'll actually cover how to create these phylogenetic trees, or what determines whether species are diverging and where they diverge from. But again, before we get to that, we're going to talk about the kind of the nitty-gritty anatomical features. Another component of phylogenetic trees are clades. Clades are essentially monophyletic groups. Monophyletic groups are groups that include the common ancestor and then all descendants of that ancestor. And that's exactly what a clade is. Clades are interesting because you can essentially create various clades or different monophyletic groups based on the ancestral node that you use, or the ancestral node of interest. And this concept means that you can have multiple monophyletic groups or multiple clades nested within one another. So the clades essentially form a nested hierarchy where you have some clades or monophyletic groups that are very inclusive and some that are rather exclusive. So it's kind of changing the relative scope per se, depending again on what ancestral node you're using to classify that monophyletic group on. So each of these colored shapes here represents a clade, right? So the largest clade stems from the common ancestor that's situated here. So this is ancestor number 7, we'll say. So when we're looking at this node, right, the clade encompasses all of these species of interest, right? This is the ancestor from which all of these species diverged from. So in making the clade, you have this ancestor and all of its descendants. But if you changed your scope of focus to this node here, the ancestor is now situated here at position 6, I think. Yeah, 6. And that means that this is the common ancestor, and the descendants of this common ancestor are D, E, F, G, and H, right? And then if you use this position, you create a clade that includes this ancestor and then E, F, G, H. Using this node would create another clade or monophyletic group. This would create another. This is one, and then this is one. So basically, every position where there's a node, at every node, you can create a new clade or another monophyletic group. And again, they will all nest within one another. So when we talk about species, technically, any given species is a member of multiple clades at multiple levels. It's depending on our scope of focus. So when we talk about Mammalia, right, there's several nested clades in Mammalia. So when we look at this ancestor here, this is the ancestor that gave rise to the majority of the individuals in Mammalia, plus your monotremes of Mammalia. All of these species are descendants of that common ancestor, so they all form the clade Mammalia. if we look at this node here, so here we have a new ancestor, and this ancestor gave rise to these species, right? It's not the ancestor of your monotremes, it's only the ancestor of these species. That forms the clade Thera. And then this common ancestor here forms a clade with these species, right? So these are all the descendants of this ancestor here, and they form Eutheria. And then there's another divergence here where you get the clade Carnivora, which are your canids and large cats or felids. But they're both the descendants of this common ancestor. So each one of these is a different clade that's nested in one another. A lot of people get confused when it comes to kind of distinguishing between phylogenetic trees because they forget that you can have rotation around or about a node, and that rotation changes kind of what the phylogenetic tree may look like, but it doesn't change the information that's conveyed by the phylogenetic tree. You still have the same tree. So these two examples, even though they have kind of different placement about the node that's over here on the right-hand side, it's still the same phylogenetic tree. Looks different, but it's still the same information that's conveyed. Figaro, don't eat my food. Will you stop it? So that's what I'm kind of showing down here. No, it is not playtime. No, I'll play with you later. Go find your squeaky toys. So if you rotate, or rotation can happen at any of these three nodes, meaning that you can kind of swap the position of 3 and 4, so you have 4 and 3. Anytime you do that, you're changing what the kind of the appearance of the phylogenetic tree, but it's still the same tree. It still relays or conveys the same information. So the information relayed by this tree, even though it looks different, is the same information relayed by this tree, this tree, this tree, and this tree. And the same goes for all of them. So you can have rotation about these nodes. It's just like when we talk about going into talking about like isomers, when we're talking about molecules, you can have natural rotation about a single bond, even though it kind of changes superficially the appearance of that molecule, it's still the same molecule. Again, you just have free rotation about that single bond. It's the same concept as this. You can rotate around these nodes, but you're still going to have the same tree. The same applies for kind of when you draw phylogenetic trees. There's two types of or forms of trees as far as format that can be used when you're drawing the phylogenies. They both look different, but they are equivalent ways of displaying information. They both relay the same information. One is tree format, the other is ladder format. So tree format, the phylogeny is kind of depicted as these sets of nested rectangular brackets. With ladder format, it's more of a slanted structure. So they look different, but they are both conveying the same information. That remains unchanged. It just looks different. All right, so let's talk about some of the different groups or types of groupings that you'll find in phylogenetic trees. The major one being monophyletic groups. So monophyletic groups are kind of these naturally occurring groups, and they include the common ancestor and all descendants of that ancestor. So here, looking at this ancestor here, forms a monophyletic group that includes H, I, and J. Right? All of these species are descendants of this ancestor, and that forms a monophyletic group. If we took it back a step further and used kind of this marking or this branching point for the ancestral node, we could form a monophyletic group that would include G, H, I, and J. Right? All of these are descendants from this common ancestor. So again, this is the group that most often occurs and we most often talk about. Another type of group are polyphyletic groups. Polyphyletic groups are very odd and they are no longer used in modern systematics because they do not adhere to the kind of the rules of forming proper clades. A polyphyletic group is one that does not include the ancestor nor all of the descendants of an ancestor. So it's almost like a random grouping of species per se. So the example given here is the grouping of E, F, and G. Right. So this is a polyphyletic group because it contains species G, which is not a descendant of the common ancestor for E and F. So again, it's more of this disjointed group. A good example is with pachyderms. So this is a partial phylogenetic tree of Mammalia that shows different monophyletic groups, right? So elephants, manatees, and hyraxes, they all form one monophyletic group. Tapirs and rhinoceroses, that's another monophyletic group. And together with the addition of horses, so using kind of this ancestral placing here, creates a third monophyletic group. But elephants, rhinoceroses, and hippopotamuses are a polyphyletic group. Right? These are pachyderms. Pachyderms are large mammals that have thick skin. And if you grouped these three species based on that kind of anatomical grouping or morphological grouping, you would have a flawed tree, right? It's more of this adaptive or an analogous trait rather than a trait that arises from an immediate common ancestor. So if you try to group those three together, you get a polyphyletic group. And because it exists as a polyphyletic group, pachyderms is no longer used as a type of group for creating phylogenetic trees. Again, it's more of just a similar characteristic that they all share. The third group are known as paraphyletic groups. Paraphyletic groups include the common ancestor, but not all of the descendants of that ancestor. So here you have a paraphyletic group because we're working with this ancestor here. So we're looking at the species or the descendants of that ancestor, but by grouping only B, C, and D, we're excluding A. So because we're excluding A, even though it's one of the descendants, we have a paraphyletic group. If A was included, we would have a monophyletic group, right? So you can think of paraphyletic groups as being a monophyletic group that's missing some of the descendants. Again, it has the common ancestor, but not all of the descendants. A really great example of this is in talking about fish. So when we say fish kind of as this organismal grouping, it seems pretty natural. We would kind of naively assume that to be a monophyletic grouping, but when you actually look at this group fish on phylogeny, you can see that the common ancestor that all fish descend from is the same common ancestor from which all vertebrate tetrapods extend from. Now, we would not call these vertebrate tetrapods by any means fish. So when we talk about fish, we're talking about a paraphyletic group, right? Because we're talking about or recognizing the common ancestor, but only some of the descendants of that common ancestor, right? So we're only talking about a subset of all of the descendants that need to be recognized. Again, if we were talking about all of these in general, this would be a very large clade, a very large monophyletic group. But because we're only talking about this area and excluding these descendants, we have a paraphyletic group. So those are the three major group types. This is another image to kind of suit your needs and help kind of map out the two different groups. Now, how do we actually make these trees? How do we actually determine where these species fall on a tree and where they diverge from, or which species diverge from which ancestor and where, etc. The whole process of making trees, we call it cladistic analysis, and it follows what's known as the parsimony principle. The parsimony principle states that the simplest explanation is the best explanation in scientific events. So you kind of use the simplest means possible to create these phylogenetic trees, right? The most parsimonious tree is the one that requires the fewest evolutionary events, meaning the fewest appearance of shared derived characters. And in making most of these trees, you'll use genetics, but you can also, so for this one, it uses some of these more anatomical structures, and organisms or species are kind of grouped together based on these shared derived characters. These are known as synapomorphies. So synapomorphies are unique characteristics that distinguish species and separate them from other species. So what you do and kind of how we make these, you take your characters, whatever character list you have, and you take the taxa of interest, and you write out, okay, what the characters are and which ones have what characters. And then you actually recode that table into this binary numerical table comprised of zeros and ones. That's why it's called binary, where 0 represents the absence of that character, 1 represents the presence of that character. And you can build your tree from bottom to top based on the number of differences, or the number of your taxa, or the percent of your taxa that character appears in. So to kind of start your tree, you always look for a character that's either in, or you look for a taxa that either has one or none of the characters of interest. So for this tree, that would be our lancets, right? These are the outgroup. So lancets have none of these characters that we're using. The characters we're using here is the vertebral column, or your spinal backbone, jaws, the tetrapod limb structure, so 4 walking legs, amniotic eggs, and hair. So lancets have none of these characters that we're using. So they serve as the outgroup, right? So they would be the first kind of branch to diverge from the main phylogenetic branch, right? So this is rooted, so here's the root. This represents the ancestor of all of these individuals, assuming that they all come from a common ancestor. So lancets are different from all of these other species that we're interested in because they don't share any of those characters, right? They don't share any of those synapomorphies. So they're an outgroup. So that's, again, kind of your base. You can then determine what divergence event comes next based on what species has only one, so in this case, only one of the characters that we're interested in. In this case, it's the lamprey. So we can say that the lamprey, tuna, salamander, turtle, and leopard, they're all related to one another because they possess this synapomorphy, being the vertebral column. But lampreys are different from your tuna, salamander, turtle, and leopards because they do not have jaws, right? So jaws serve as the synapomorphy or the unique shared derived trait for your tuna, salamander, turtle, and leopard. And that's what allows us to recognize lampreys as kind of this second species, or recognize that they occurred at this site in evolutionary history. So again, your tuna, salamander, turtle, and leopards, they're all related because they have jaws. Now, how are they different? Well, we can say that tuna are different from salamanders, turtles, and leopards because they don't have your four walking legs, right? So the four walking legs becomes a synapomorphy for these three species. Because tuna does not have those four walking legs, we can say that it's not related to those three. So that's why you have a divergence event at this placement. We have another divergence right after this synapomorphy, so right at this site, that shows salamanders are different from turtles and leopards. Salamanders are different because they do not have an amniotic egg. So the amniotic egg is the synapomorphy that relates turtles and leopards, and then leopards are different from turtles because they have hair. So when you're building the tree, you're going from kind of the largest number of shared derived traits to the smallest number. So the base, as you move from the base to kind of the distal nodes of your phylogeny, you're going from characters that occur in most of the species to those that are more unique or in less of the species. So it's essentially the same that we do with the generating trees from the nucleotide sequences, right? So every nucleotide or every substitution that occurs in the nucleotide sequence puts distance between two organisms. So you would start the tree, kind of the most base divergence would be between the kind of two species that have the greatest number of nucleotide substitutions. So if we were give an example, we could say like if we're making a phylogenetic tree, we would use the first divergence would be, say, between humans and yeast, because there's 53 or 56 amino acid substitutions between the two. And then humans are most closely related to chimpanzees because there's only one amino acid difference. And after chimpanzees, kind of moving down the tree, you would have like rhesus monkeys and going on from there. So it's all determined based on the number of differences that occur when you're using these characters. It's based on the number of species that share that character. So as you go from kind of the ancestral to more present day in your branching pattern, you're moving from most common to least common. Traits that are highly prevalent or present in your species to traits that are very unique or kind of retained to single species. So that's how, again, how you make these phylogenetic trees. So here's another example. Again, each of these blue dots represent the common ancestor or divergence event, and you are distinguishing these relationships based on synapomorphies, these shared derived traits. So species will, or certain groups will have these shared derived traits. If they have a shared derived trait, that means they stem from a common ancestor that had that trait. So here, this was created just like the previous, not this tree, but this one here, using that the notion of synapomorphy. So again, each ancestor has this shared derived trait that is present in all of its descendants, right? So that's how we know that these individuals branch from a certain individual. If they have that shared derived characteristic, they're related in that aspect. So here we can say, looking at the 11 clades of chordata, when we talk about kind of going a step back, we talk about deuterostomes. You have your echinoderms and then all of your chordates. We can say that, yes, they all arise from this ancestral deuterostome, but your chordates are kind of separate. They're a separate clan. They are different from echinoderms because they possess the notochord. Notochord being that primordial cartilaginous spinal column. It was very flexible and useful for swimming. Because echinoderms lack that synapomorphy, we can say, okay, a divergence event occurred at this ancestor. We know they share this ancestor, but because echinoderms don't have this synapomorphy that's present in all your chordates, we know that there was a divergence event here. So at this ancestor, you had the divergence of the two groups to form your echinoderms and then your chordates. And then you can further kind of subdivide all the groups of chordata based on those synapomorphies, right? So we can we form tetrapods, or we say that tetrapods are different from your lungfish. So they both arose from this common ancestor because they all have this synapomorphy, but your amphibians and reptiles and mammalia are different from your lungfish because they have limbs with digits, right? So because they have this synapomorphy, or they all share this synapomorphy, and it's not shared with lungfish who come from the same common ancestor, we know that they are two separate groupings. So that's, again, the concept that's being used here. Each one of these represents, again, a divergence event where you're coming from a common ancestor that has some synapomorphy that's present in the species of interest. And then you can kind of distinguish between those species based on other synapomorphies. So here, your lungfish and all tetrapods they come from this ancestor, and the synapomorphy that's linking all of these groups together are lungs, right? So because they all have lungs, they all came from this common ancestor, right? But we know that lungfish are not tetrapods, right? So we separate the two because your tetrapods have this synapomorphy that makes them uniquely different from your lungfish. So because tetrapods have tetrapod limbs, they are a different group, or they are different evolutionarily from your lungfish. So at this ancestor, you had the the divergence of these two groups. And you know it occurred here because they both have the synapomorphy that is retained in this ancestor, but lungfish do not have the synapomorphy of the next ancestor that's linking the groups. So we can move on to tetrapods. So tetrapods all arise from this common ancestor listed as number 2, because they all have the synapomorphy tetrapod limbs. Right? So because they all have this shared derived trait, they are all related to one another, or all derived from the same common ancestor, and are grouped as tetrapods. But we can go on and say, well, there was a divergence event here that separates amphibians from this other grouping of species called amniotes. And the reason why we're able to do that is because your amniotes all share a derived characteristic known as the amniotic egg, and amphibians don't. And amphibians also use gas exchange, or have gas exchange occurring across the surface of the skin. So that makes them very different from this group, because they do not share the next derived trait that is found in all of these species. They are recognized as having diverged at that kind of last common ancestor for all tetrapods. So next divergence event, right? So here we have the ancestor for all amniotes, right? Amniotes are your mammals, lizards, and birds. They all have the amniotic egg. But we can say, okay, well, at this common ancestor, yes, this is the common ancestor for all those species, but at this site you had the divergence of mammals from these other species, because they do not retain the synapomorphy that links or connects or groups your lizards, crocodiles, ostriches, lizards, crocodiles, and birds. So mammals are different from these guys because they are viviparous, and they have mammary glands. So they produce milk. They're able to lactate. They also have fur. So those differences distinguish them as being separate, or distinctly different from this grouping here, which enables us to indicate that there was a divergence event at this ancestor. So this is the shared ancestor because of all amniotes, because they all have amniotic eggs, and that was the synapomorphy that links them from this ancestor. But at that time, or from this ancestor, you had the divergence of mammals from these other species, because mammals have milk production and are viviparous. These other species are oviparous. Now, kind of again moving on in our phylogeny, your lizards, crocodiles, and birds, they are all related or all arrive from this common ancestor. And we know that they're all derived from the same common ancestor because they possess this synapomorphy, which is retained in the ancestor. The synapomorphy here would be the opening in the side of the skull, right? That's what makes them, or they're also recognized as diapsids. So all of these are diapsids because of that synapomorphy, or that homologous characteristic that's retained from their common ancestor. But we can say that lizards and snakes, your reptiles, are different, or they diverged at that ancestor from your crocodiles and birds, because they have their differences, right? So first of all, your lizards and snakes, they don't have a gizzard, right? So that makes them uniquely different from these guys. They also do not have the shared derived trait that you find kind of synonymous between your crocodiles and birds. So because they do not, or because your reptiles do not have, as far as your lizards and snakes, because they don't have this synapomorphy, we can say, all right, so at this common ancestor, the common ancestor for lizards, crocodiles, and your birds, you had the divergence of lizards and snakes from crocodiles and birds, because they don't have the synapomorphy that's shared by crocodiles and birds. Crocodiles and birds, you can then say, all right, their common ancestor is right here at position 5, because they all share the synapomorphy, or the derived trait, where they have skull openings in front of the eye and in the jaw. So that's the synapomorphy that links these 3 groups, or more specifically 2 groups. So it links your crocodiles and birds together. But we can say that at that ancestor, yes, they're all linked to that ancestor, they're all related, based or stem from that common ancestor. And at that ancestor, you had the divergence of crocodiles from birds, because crocodiles have secondary palate or plate in their mouth, whereas your birds have feathers and toothless beaks, right? So that's what separates them. That's how we know there was a divergence between those groups at this ancestor. Now, your birds obviously are related, or stem from this ancestor, because they shared the synapomorphy of feathers. So feathers is a synapomorphy that links your ostriches and other bird species together. But we know at that ancestor there was a divergence event, right? So we recognize a divergence of ostriches from these other bird species, and that divergence is based on their ability to fly. So ostriches, here you have your flightless birds, and here you have birds of flight, right? So they all arise from the same common ancestor. We know they arise from the same common ancestor because they have or retain this shared derived trait, or synapomorphy. But we also know that at that ancestor, they diverged from one another because of the differences that I noted. So that's, again, kind of walking you through making a phylogenetic tree. Again, going from the kind of least number of differences to most. And when I say that, you're going from kind of starting at the base of your tree, or the root, being where you kind of have a shared derived trait that is retained in all but one species, and then moving forward in your traits, going through kind of a numerical succession, that as you move forward or along your phylogenetic tree, this specific, or the shared derived trait becomes more and more unique to an individual species. So you're basically going from a trait that's, or synapomorphy that's highly present in all of your species of interest, building up, or following up to one that is retained in only one species or a small group of your species. So that's, again, how you make your phylogenetic trees. I hope this video helped. If you are having any difficulty with this topic, I know some people get kind of really wrapped around the axle when it comes to phylogenetics and making these phylogenetic trees. But I hope this video helped shed some light on that matter, that you can walk away with a slightly better or clearer understanding of how to do so.